Sir, good morning, sir. The registered garu, shall we start?
Okay, Dhananjay garu.
Okay. Sir, good morning, all of you. Welcome to Dr. YSR Horticulture University, Venkataraman West Godavari District, Andhra Pradesh, for the 22nd Global Connect series, which is arranged by Dr. YSR Horticulture University for the betterment of faculty and students by giving the international seminars by reputed international scientists from different countries. We have conducted 21 Global Connect series. This is 22nd Global Connect series in collaboration with Delaware State University, USA, on enhancing strawberry resistance to gray mold through fungicide alternative studies, advanced phenotyping, and genetic research. So the topic today, and respected Honorable Chairman and Vice Chancellor, Dr. K. Gopal Garu, with his dynamic and visionary leadership, we are conducting 22nd Global Connect series. Thank you for giving the permission to conduct 22nd Global Connect series by Director of Industrial and International Programs. And today, the guest speaker, Dr. Amarnath Reddy, the postdoctoral research associate, Department of Agricultural and Natural Resources, Delaware State University, Dover, USA. Welcome, sir. Welcome to Dr. YSR Horticulture University. Dr. Amarnath Reddy garu. And
thank you, sir.
Yes, sir. And all university officers, Dean of Horticulture, Dr. Naram Naidu garu, Director of Research, Dr. Madhavi garu, Director of Extension, Dr. Govind Rajul garu, and PG Dean, Dr. K.T. Venkatramana garu, Dean of Student Affairs, Dr. Surya Kumar garu, Registrar, Dr. B. Srinivasulu garu, University Librarian, Dr. Ramanandam garu, Controller of Exams, Dr. Prasanna Kumar garu, Controller, Dr. Srinivas Subramanian garu, State Officer Ramana garu, Director of Planning and Monitoring Cell, Dr. Vijay Padma garu, all Associate Deans, all Heads of Research Stations and KVKs, and all Principals of Polytechnic Colleges, all scientists, teaching and teachers, scientists and students. Welcome to this 22nd Global Connect series. Good morning once again, all of you. And today the rapporteurs are Dr. C.H. Nalini, Assistant Professor, Entomology, College of Horticulture, Venkataramanagudam, under Dr. S.B. Ramya Lakshmi, Assistant Professor, Agriculture Economics, College of Horticulture, Anantarajpeta will act as rapporteurs for today's program. And before going to speech by the speaker, just I will have a brief introduction of Dr. Amarnath Reddy garu. Sir is native from Kadiri, from Anantapur district, Rayalaseema region. And Dr. Amarnath Reddy garu obtained a master's degree in botany with a specialization in plant molecular physiology from Sri Venkateswara University, Tirupati. He pursued his PhD program in botany at Sri Krishna Devaraya University, Anantapur, and as part of research conducted at the University of Agriculture Sciences, GKVK, Bangalore, India. His PhD research was focused on the development of rice transgenics using the multi-gene staking approach for improved herbicide tolerance and characterization of biotic stress tolerance genes in croplands. And since December 2016, he worked as a postdoc with Dr. Mithila Jugulam and Krishna Jagadish at Kansas State University, USA. His research was focused on screening and characterization of croplands and weeds for herbicide resistance, cold and high night temperature stress tolerance, and understanding physiological and molecular mechanisms Currently, the research in Dr. Kalpalata Millai lab at Delaware State University is on development of genetic resources for climatic resilience in small fruit horticulture crops such as strawberries and blueberries crops, industrial hemp and sugar maple. And SAR has published more than 35 research publications in national and international reputed journals. Welcome, sir. Welcome to Dr. YSR Horticulture University. And with this brief introduction, I invite Dr. Amarnath Reddy Garu. Thank you for accepting our invitation from Dr. YSR Horticulture University for giving speech on this topic. Thank you, sir.
Thank
Dr.
you.
Amarnath Reddy Garu. And you can put that now.
Okay, sir. Thank you.
Presentation. Can share
Sure,
your
sir. A minute. I'm going to share my PPT.
Yes, sir.
Now, are you able to see my screen, sir?
Yes, sir. Audible, visible also. Excellent. Please
Okay.
go ahead.
Yeah, sure. Thank you, Dr. Dhananjay Rai garu, for a nice introduction and also for giving me this opportunity to you and also Honorable Vice Chancellor and also all the faculty and staff at the Dr. YSR Horticultural University. Thank you. So today I'm going to talk about like enhancing strawberry resistant to gray mold. 1 minute. Okay. So we generally use here mostly WebEx, but Zoom little bit. 1 minute. I'm.
In slide mode, please.
Yeah, that is what I'm checking, like how to go to slide. Thank you.
Otherwise, you share to my WhatsApp. We will share from here.
Okay, sir. So in the view mode, I need to only speaker. I'm just changing my.
Yeah, no problem.
Okay. So yes, today I'm going to talk about enhancing strawberry resistant to gray mold through fungicide alternative studies, advanced phenotyping and genetic researchers. So I'm currently working as Dr. Dhananjay Rai mentioned, postdoctoral associate at Delaware State University in Dr. Melmai's lab. So as most of the people, the strawberry is like now a widely consumed horticultural crop in the worldwide. But if you know that like what we are eating, the strawberries are like currently consuming, or most of them are hybrids. So the hybrids, like if you wanted to know, like little bit curiosity, how they came from and where they came from actually. So now that what we are seeing, the garden strawberry is the one we are like consuming now, but this came from the two different wild strawberries. One is from Chile, South America, and another one is from Fraseria virginiana from North America. One is from US and another one from South America. So they took these two genotype or whatever this wild genotype to the Europe, and there they developed this variety. Now whatever widely we are consuming is garden strawberry, Fraseria ananasa, is developed from these two wild varieties. So when you see that like wild varieties are like the fruit size is very small and also leaf size, if you see that all the characters were like through the selection they selected and now developed this garden strawberry. So why the strawberries are very important, why people are very like, they most of the people are loving strawberries is because of their flavor and also taste. Other than that flavor and taste, also strawberries have like a really high nutritional benefits such as vitamin C, antioxidants, vitamin B, and also other vitamins. Also, more antioxidants, like anthocyanins, and also rich in potassium. Why? Since they have this kind of nutritional values, they also gives like good human health related traits when you consume, like they can prevent like cardiovascular diseases, and also they have anti-cancer properties, and also they play major role in reducing aging. Also, they induces the immunity. So coming to the strawberry production worldwide and market size, if you see uh, worldwide production, China is in the topmost producer for strawberry production, and United States is the top second most producer of strawberries. So there are different countries varies, and but most of the times, if you see year wise, China and United States are like always in the top in terms of production. So if you see the global market report for strawberry, the market size is steadily increasing. So if you see in 2024, it is 15 billion. They are expected to reach to 16.5 billion in 2025. And also the growth, like if you see the map, the prediction is saying that by 2029, it will be reaching to 19 billion market size. So that suggests that like the compound annual growth rate per year, it is like almost like 4.4%. That means it's steadily increasing. So that shows that the market is increasing, means the demand is more. So coming to the US and United States is, even though like top second most producer, the production is only limited to few states. If you see, like 59% of the strawberries comes from California, and 11% of strawberries comes from Florida. So these two are like a kind of tropical weather, kind of temperate kind of weather you can see in the United States. So, but like if you see, the 75% of the strawberry production comes from like either western states or southern states. So even though there are other states also produces strawberry, in terms of percentages, they are less than 2%, or some of them are 1%. They did not mention in this map if they are lesser than 1%. So if you see that even within the states, the major, like the intensity of red color shows here the major strawberry growing areas. It's limited to only few areas. So coming to India also, there is not much statistical data available for in terms of strawberry production. But some studies shows that in the predicting that in the future there will be high market size or market demand for the strawberries. By 2029, there will be 3.4%, and they are expecting that 2,400 US million dollars in the future for market size. Strawberry demand in the United States. See, even though United States are top second most producer, the production is the demand is higher than the production. So now, even though it's producing a lot of strawberries, but people still they need more. So that is why they are exporting from other countries, near neighboring states, like countries like Canada or other uh, nearby countries or South America. So that indicates that there is higher demand than the production within the United States or North America. If you see per capita consumption for strawberries in the United States, in the 2000 it was like 4.8. Now if you see 2020, like almost like 8.5. Now steadily it's increasing. That means like the consumption rate is really high in the United States. That indicates that the more demand for strawberry production. So even though it is top second most producer, but still there is a higher demand for the strawberry, because it's not only for consumption. If you see, strawberry industry also big in US in terms of producing any like processing products, like flavor for strawberry, like it's a big industry in the US, and also they frozen a lot of strawberries and they will export to other neighboring states or neighboring countries. So that's why it's like higher demand in United States. So even though it has higher demand, but strawberry production also has major challenges in US. So strawberry being a weak plant, and that is very sensitive to biotic and abiotic stresses. So when the plant is stressed, it experiences more diseases. So since it is a weak plant, as I mentioned, when if it is dry or if it is temperatures too hot, it will become more sensitive to biotic stresses. So the major biotic stresses are the fungal diseases. Among the fungal diseases, like if you see here, are any other diseases, leaf blight, leaf spot, anthracnose, and powdery mildew are the major diseases. Among all, compared to the gray mold, is the major or devastating diseases. They call it because it's a necrotrophic fungus. It throughout the cycle of the plant cycle, it infect the plant. So that's why they call it a necrotrophic fungi and devastating disease. So if you see here in the life cycle, so the source for the fungal disease either by dead parts, also through the sclerotium. So it can infect at any stage of the cycle. The primary infection generally occurs through the leaf, from the leaf, through the flower. From the flower, it will infect the fruits and it will continue the cycle. The fruit, again, dead bodies, that whatever the conidia has produced through cycle will continue, again infect the fruits, or it can infect the leaves. So the cycle will continue. So that is why, like any stage of the plant, you can see the gray mold infection on the plants. But even in the vegetative stage, it will be latent form, but you won't see the severity of the disease. But when it comes to the fruit, the symptoms are significantly high. But the pathogen affects fruits in the field. So once it infected in the field, when you harvest, the plant or fruits may look good, but when you store, you can see the fungal growth, or even while transport and on the market, you will see that is the major disadvantage of this gray mold. And consumers also doesn't like when you see the fungal growth on the fruit. That is why it is considered as one of the devastating diseases for the strawberry. Since it has like a lot of this fungal infection, and people or consumer losing the demand because of this infection, so they apply a lot of pesticide like fungicides, like they apply before harvest and also after harvest. So the widely usage of fungicide has become prerequisite in strawberry production and storage. So wide use of pesticides because is happening in the strawberries, and see if you see that lot of labeled pesticides used for strawberry. So but the wide usage of fungicides leading to the evolution of fungicide resistant in the fungus, and also once the fungus or botrytis developing the resistant to this fungus, is they are no more effective on this fungus. So the fungal strains are developing resistant. If you see the frequency of fungicide resistance in botrytis, see, so most of this already this gray mold or botrytis already developed resistant to the most of the fungicides. That indicates that most of the fungicides what have been labeled for strawberry is not effective no more in the strawberry production. So since they are not effective, farmers started spraying a lot of pesticide. And if you see in the 2024, the strawberries has been reported 30 dozen fruit. If you see in the 30 dozen fruits, the strawberry place in the first place.
Please.
If somebody can mute them, sir, or can I continue?
శాంతకుమారి గారు,
Pawan.
శాంతకుమారి గారు, ప్లీజ్ మ్యూట్ యువర్. హలో,
Madam Shantakumar garu. Pawan. Okay. Sir, proceed.
Okay, sir. Thank you. So, due to the higher pesticide usage, the strawberries reported in, even though they are like really good for health in terms of they have nutritional benefits, but they reported as one of the 30 dozen fruit they listed strawberries in the top position. That means the higher usage of pesticides. So due to this, there are rising concern of environment and also human health. So now people are like really concerned to consume the strawberries due to this report. In 2024 also there was a report in 2023 as well. So it's now the researchers and also like consumers are looking for less pesticide or organic strawberries options. So now as I mentioned, the researchers and also consumer demand is looking for fungicide alternative or less harmful chemicals, because farmers they cannot grow, because like now that most of the strawberries are resistant to this fungus, but they cannot control. Still they have to adopt this because fungicides, even though they are harmful, but if you use them in right amount and right combinations, they can control and be beneficial to the farmers. So that's why people are looking for fungicide alternatives or less harmful chemicals, or identifying the resistant germplasm is another option. So if researchers can develop fungicide resistant strawberries, that will be useful for the farmers, also consumers. So with this, our research like focus on our objective was study the fungicide alternatives to control the gray mold and to develop the resources for botrytis resistant in strawberry through advanced phenotyping and genotypic research. So among these, like we like are like a lot of people reported that potassium silicate is one of the it's like a chemical, but they reported it is like one of the essential nutrient element in the earth, so that is required for the plants. So that's why it's not considered as a chemical, even though we are supplying in the form of chemical. This is nutritional or beneficial element for the plant. So because it strengthens the plant physiological or morphological structures, and also it protects from the fungal and also other diseases and also environmental stress. So we conducted one high tunnel research. The strawberries grown under high tunnel using one of the field kind of structure here, the high tunnels at DSU, like our Delaware State University Outreach and Research Center at Smyrna, Delaware. So this potassium silicate, so we sprayed to the strawberry plants. As I mentioned, this is considered as organic. So that is why we use this chemical to test on this alternative, the fungicide on the strawberry. So this international the pesticide national that institute nutritional institute reported this potassium silicate as a organic compound. So that's why we tested this by treating on different concentrations. So 2 ml per gallon, 3 ml per gallon, and 4 ml per gallon concentrations, and control without any potassium silicate. Then we measure that once we spray the potassium silicate, the potassium silicate accumulation in the plant parts such as roots and leaves, we have checked the concentration using the scanning electron microscope and also spectrophotometric method, and also we assessed this potassium silicate antifungal activity using PDA media. Because even though they reported, but there was no comprehensive studies how this potassium silicate or what dose is effective kind of studies in strawberry. That's why we have chosen this research to test this chemical on strawberry. And also we conducted one post-harvest fruit assay to check that potassium silicate, how effective on fruits. So coming to the results, so potassium silicate treatment. So if you see that we measured marketable yield and non-marketable yield of the strawberry, and if you see the different treatments effect, and also here we measure the plant height and also plant width. So among all the treatments, so we tested on two strawberry cultivars. One is Flavor Fest and the one is Rutgers Scarlet. These two are the commonly cultivated strawberry varieties in the northeast of the United States. So among all the strawberry concentrations, if you see the 2 ml per gallon treatment showed significantly or better in terms of increasing the yield. If you see in both the genotypes, So the yield compared to other treatment was significantly high, and also the plant height was also high, and also the plant width was significantly high compared to other treatments, suggesting that 2 ml per gallon treatment is better in terms of increasing the plant physiological functions and also the morphology of the plant and yield. So you can see here the clear pictures, different treatments, 0 ml, 2 ml, and 3 ml, and 4 ml. So if you see the plant width and 2 ml treatment is significantly high. So that is what we observed. And also coming to the fruit yield and size. So compared to all other treatments, you see the 2 ml treatment in terms of fruit size and yield is more compared to other treatments. The size of the fruits are less. So suggesting that 2 ml treatment significantly enhance the fruit size and yield. So coming to the silica accumulation or potassium silicate accumulation in the plant parts. So here this is the pictures was taken, or microscopic pictures from the scanning electron microscope. So the first picture, the first panel shows the black and white. So here if you see that if there is a white precipitation is more, that means that silica concentration is high. If you see that the white or dumbbell shaped structures are more in 2 ml treatment, suggesting that silica accumulation in the 2 ml treatment is more compared to other treatment. That is why plants are maybe possibly showing better response, because the accumulation of the silica is more in the 2 ml treatment. And in the second picture, this overlap of potassium and silica both. So if the picture shows more orange color, that means the more silica. So if you see compared to other treatments, in the 2 ml treatments, the dumbbell shape structures in the leaves showing that more structure and the crystals are more, that suggesting that 2 ml treatment is more effective. If you see in the multi-layered picture, so silica accumulation, because the orange color is high, that is suggesting that silica is more, and overlap of all elements like potassium, silica, and also other elements in the plants. So if it is still more orange color, means that is more silica. So compared to, see here the picture is showing pink color, here is pink, and the structures are like showing small, but in the 2 ml treatment the structures are strong, and also you can see that more orange color, that suggesting that more silica accumulation and upon 2 ml treatment. So that EDS spectra of the same electro energy dispersive X-ray spectroscopy coupled with this scanning electron microscope. So here if it is silica peak, if it is high, that means the peak area indicates the concentration. If peak area is high, that means silica concentration is high. So here if you see in the no silica treatment, the concentration is low. So when they treated with potassium silicate, see the peak area is increased and the concentration also significantly increased. If you see here, the silica concentration was 22%. If you come here, the silica concentration is 32. So from 22, 32, 10% is increased upon 2 ml treatment. So compared to all other treatments, the peak area significantly higher at 2 ml, and also the weight percentage is more at the 2 ml treatment. So this is in the roots. So we sprayed potassium silicate on the leaves actually, then we measured the potassium silicate concentration both in the leaves as well as the roots. So coming to the roots, see the structures of silica is not that strong enough. I mean, like more not concentrated. Even the molecules are not strong. And here also you see that compared to all other treatments, the 2 ml, the intensity of that color, still the orange color is more compared to others, suggesting the, sorry, in 2 ml treatment in the roots is less. See, if you see here that 3 ml, 4 ml, the structures are bigger than the 2 ml. That means the whatever we sprayed upon 2 ml treatment, that accumulated more on the leaves than the roots. But in the other treatments, it translocated to the roots compared to the 2 ml treatment. So the structures are bigger in 3 ml and 4 ml compared to 2 ml treatment. So also the peak area, also see in the leaves, the peak was really high in the 2 ml, but here it is not much. It is similar to other. But if you see that in the 4 ml treatment, the peak area is bigger than 2 ml and 3 ml, suggesting that 2 ml treatment mostly accumulating, making this aerial parts like leaves and other outer parts more strengther than the roots. So coming to, and also we measure that silica concentration in the both root and shoot, I mean leaves and the roots. So if you see that upon 2 ml treatment, the silica concentrations were, excuse me, significantly high at 2 ml treatment compared to other treatments. But when it comes to the roots, the 2 ml treatment showed lower or concentration of silica, suggesting that mostly the 2 ml treatment, that whatever we are spraying is becoming very effective and accumulating in the plant and the aerial parts of the plant and making it strength compared to others. So then other than that, also we wanted to study why this potassium silicate becoming effective in the plants. So also that in other crops, they reported that potassium silicate is effective on controlling other funguses, but nobody studied so far on like at least constructive or comparative or comprehensive study what concentration is more effective on reducing the fungal growth. So we were curious and wanted to study if this potassium silicate can also reduce the fungal growth of the Botrytis cinerea or gray mold. So we studied using different concentrations of potassium silicate 0 ml, 1 ml, 2, 3, and 4 ml, and 5 ml. So if you see that when we added the potassium silicate to PDA media and we placed the Botrytis scenario fungal strain, at all the concentration, day 1 and day, up to day 1, day 2, there is no difference. But when it comes to the day 3, after inoculation of this fungus, if you see at 2 ml treatment, the fungal growth is not reduced fungal growth. You can observe at 2 ml treatment on day 3, and also on 4th day, you can see the significant reduction in the potassium silicate treated at 2 ml treatment. And also on the 5th day, if you see that significant reduction in the radial growth of the fungus compared to other treatments, suggesting that 2 ml treatment maybe have some effect on fungus, maybe because at that concentration, it maybe it's absorbing, that is why, or it is maybe in the form of it's like less effective on fungus. Also we don't know yet, but we need to like do more studies to confirm this, or constructive studies needed to confirm this. So, and also we were curious, since after finding this, so we wanted to test this potassium silicate really can affect the fruits if we treat, or we wanted to test. So we tested that potassium silicate effect on post-harvest strawberry fruits. We challenged with the Botrytis scenario here. So in here, if you see in the first 0, we for this study, we selected 2 strawberry genotypes. One is Ruby June and Chandler. So these are the like cultivars that grown widely in the commercial in the United States. So in the first picture, we did not treat any potassium silicate or any fungus. So you can see that normal fruits harvested from the field. In the second picture, and all the fruits were surface sterilized with sodium hypochlorite. So in the first one, we just treated with Botrytis. In the here, if you see that we treated 1 ml of potassium silicate and Botrytis, 2 ml potassium silicate in this panel, and potassium silicate 3 ml, 4 ml with the Botrytis scenario. So if you see here, compared to all other treatment at 2 ml for treatment, we still see the reduction in the growth of this fungus compared to other treatment. Even the 4 ml, see the fungal growth is high. Even 1 ml it is high. Even 3 ml the fungal growth is high compared to 2 ml treatment. So that's suggesting that maybe this is more effective in terms of absorption and translocation to the aerial part of the plant. So if you here we calculated percent injury of the fruit. So if you see at a 2 ml treatment, the percent injury is less compared to all other treatments. So suggesting that because it's not only fungal growth, also the effect, the percent injury based on the fruit softness, and also that the effect here, if you see, even though here fungal growth is more, the fruit may not get damaged here, but here see that fruit damage is more. So for example, here also the black spot, the softening of the fruit, all factors need to be considered in terms of calculating the percent injury. So then we were curious why this potassium silicate is being very effective in the plants, what kind of genes or molecular mechanism playing a role. So with that curiosity, we wanted to test some potassium silicate transporter genes and their expression. We have chosen one of the strawberry cultivar that from this here, that Chandler variety, and we studied this expression of these two potassium silicate transporter genes. So if you see that the potassium silicate transporter genes are upregulating a 2 ml treatment compared to other treatments. So one is here ARSP, one potassium silicate, and another. They both are aquaporins, and one is aquaporin and one is transcription factor of this potassium silicate transporter gene. So you can see that at 2 ml treatment, both the genes are significantly upregulating compared to other treatments. So that possibly suggesting that this potassium silicate at 2 ml treatment, why it is because of the effectiveness of this transporter genes, it is really highly accumulating in the aerial parts and strengthening the plant aerial parts and also bringing the botrytis resistance. So coming to the conclusion of this potassium silicate study. So the potassium silicate significantly affecting the plant width and also the fruit yield compared to the control. And also that botrytis scenario growth is like high in the control, but this potassium silicate is reducing the radial growth of the fungus because of the effectiveness of the treatment. And also coming to the accumulation of the silica, it is more in the leaves at 2 ml treatment compared to the root parts. So that is why it is strengthening the aerial part and also bringing the more fruits and yield. So coming to the post-harvest treatment, the potassium silicate is significantly effective in term compared to control because treatments are reducing the fungal growth, especially 2 ml treatment. And why they are like effective, this treatment is because of the upregulation of the potassium silicate transporter genes. So, and another study, like this is our fungicide alternative study. Another study we did for evaluating the strawberry germplasm for gray mold fungal resistance. So, if you see here, so methodology we did, we screened the germplasm using the phenotyping and the morphophysiological characterization. And coming to the genotype, so we did like QTL genome-wide associations and identifying the candidate genes. So this is our genotyping study. So basically, we evaluated the germplasm and characterized for the gray mold fungal resistant genes or traits in the strawberry germplasm. For this, we used like total 250 germplasm, but in-house we had 150, and remaining germplasm we adopted from USDA. So, and also coming to the like characterization, morphophysiological characterization, we did scanning electron microscope and also chlorophyll index and using these handheld devices. And also we this is the leaf disc assay that we standardized in in-house method that we adapted to screen the germplasm and also fruit assay. And coming to the genotyping and validation, we did like microarray chip analysis, and also we in the future we are going to do the genotyping by sequencing, and also now we are making genome editing lines under a pipeline. So the standardization, coming to the leaf disc assay method. So screening germplasm is really difficult in strawberry because strawberry in US you can only grow, especially in terms of in coming to the like northeast, because of the winter season we cannot grow plants. So within a year we have to finish all the phenotyping. Sometimes it makes hard job. So that is why like instead of directly conducting field studies on large germplasm, if we can adopt some laboratory assays to screen the germplasm and then selected lines doing the field assay makes our job easy, and also weather will support for that kind of study. So with that aim, so we started doing like standardizing different laboratory methods to screen germplasm. Initially, we used whole leaf assay. So we detached the whole leaf and also we spotted them using the fungal spores suspension, botrytis. So in this method, we were not successful when we inoculated with fungal spore suspensions, we did not see any symptoms in this fungal or after the treatment. And also we like sprayed the fungal spore suspension and the leaf disc. This method is working, but the problem is this infection rate is not uniform. See, some, for example, here in some leaf disc you can still see the green, but some leaf discs are getting infected. So that is why we wanted to needed more robust or rigorous method which we can see the uniform or symptoms or more kind of uniformity in the experiment. So these two methods we were not successful. Then in our lab, without giving up, we again standardize. So in this, so we initially grown that all strawberry germplasm in the greenhouse. Then we collected the fresh, third uniform leaves and the actively growing leaves, and we surface sterilized using again sodium hypochlorite and also fungizone treatment. So we made uniform discs here. If you see that deep, and also we placed the leaf disc on the agar media 0.8%. Then we inoculated with the actively growing fungal uniform fungal discs. So 7 days, I think, for botrytis we use 7 days old fungal discs to infect the leaf discs, and we incubated them at in the dark for some time. Then we moved them to the light at 25 degrees for I think 1 week for this. So then we measured percent injury and also chlorophyll and MDA to quantifying this injury effect. So if you see here, when we did this method, our own method, see the infection rate is uniform. See, all the leaf discs are getting infected, and so that is why we stick to this method. So maybe I will show you the results in the next slide. So based on that, whatever the leaf disc method, we screened the germplasm, we identified. So if you see here, so the botrytis infected germplasm has lot of higher percentage injury, more than 75%, some even 100% injury. So this in the control, the percentage injury is less. Interestingly, some of the strawberry lines, even though showed even upon botrytis infection, less injury. So based on whichever showed more than 25% less injury, we consider them as resistant lines, and above 25 and up to 25 to 50 we consider them as moderate, and more than 50 we consider as the sensitive lines. So based on the scale, we categorize them and we use for further analysis to validate them. So, and also if you see the chlorophyll content also, the resistant lines, some of the resistant lines, they maintained higher chlorophyll content similar to control, but in the lot of that other genotypes showed higher percentage injury, or a higher reduction in the chlorophyll content. The plants which showed so for example, here you can see that A10 is the resistant line, so maintained lower MDA level compared to other. So here also if you see the 10, right, A10, 10 line here, maintained 10 here, so maintained higher chlorophyll content similar to control, that suggesting even upon infection they are maintaining more or chlorophyll content. That means these lines are resistant. So here they maintain MDA is the biomarker for the lipid peroxidation. So if MDA accumulation is more, that means membrane damage is high in the leaf disc. So for example, M11 is the sensitive line. So here if you see the sensitive line, the chlorophyll content is significantly low, and also the percent injury is high in the 11. So that means the sensitive lines showed higher percentage injury and lower chlorophyll content and higher MDA. So if you see here, the infection in these pictures is uniform. See, for example, all the 3 leaf discs got infected. This is the sensitive line. They showed more than 50% of the infection. But in control, there is not much infection. Control here also no infection, but coming to the resistant line, even upon botrytis infection, the infection rate is significantly low. So this is we consider it as a resistant line. This we consider it as a sensitive line. So like this, we characterize germplasm, and now this field level screening of this line is going on. 1 year field trials are already done. So now we wanted to, before like sharing the results, we wanted to confirm 1 more year in the field condition. So along with that, simultaneously also we did some genotyping. So 130 USDA germplasm lines we collected from USDA at Maryland, and we have some in-house in our place also. We collected all the DNA samples, and we 250 strawberry DNA samples for we extracted DNA, and now we like did microchip analysis. So now results are with us. We wanted to be using this chip analysis. We are going to do the jivas, and also we are going to identify the SNPs and also candidate genes. So this we are going to validate them, and we are going to use it for our further validation studies using CRISPR-Cas or transgenic approach to validate these SNPs and candidate genes identified this through this microchip analysis. So we are already like started our for validation of those identified candidate genes in the future. So simultaneously, we also standardized strawberry in vitro regeneration protocol for one of our identified strawberry genotype in-house. So for this purpose, we used PTO cell explants, and we successfully standardized the shoot regeneration media and also shoot elongation media, and we successfully able to get the tissue culture raised with the rooting media. All the plants now they are under establishment. So now we are ready for transformation. So to do the transformation, initially we are doing CRISPR Cas9 vector using the PDS gene. PDS is one of the like model gene for testing any other whether construct is working or not. So we are now successful. We cloned successfully into the binary vector. Now we have already confirmed the PDS gene in the binary vector. So further we are going to do the gene transformation in the strawberry, and we are going to test later our candidate genes using the same approach. So coming to the conclusion, our study like showed that potassium silicate is a very potential fungicide alternative chemical, which is organic, and it can strengthen the agronomic traits in strawberry, and also it can help in post-harvest management by doing the treatments. And also we successfully able to standardize the protocol, and the leaf discussive method. It is very robust, and we can screen a lot of germplasm using this in-house in the lab, and this is very uniform in terms of results. And also the morphophysiological approaches, whatever we characterized, some of them results are not shared here. So we have the data still, but we are still analyzing. And the genotyping and identification of resistance through our genomic study will be a potential genetic resources for crop breeding. And the standardized in vitro tissue culture protocols and CRISPR constructs will be potential sources for the strawberry crop improvement for developing the botrytis resistance. With this, I would like to acknowledge my supervisor, Dr. Kalpalata Melmayi, and our lab, Plant Molecular Breeding Lab at Delaware State University, and also our university and also our school and department for providing me this opportunity, and also the funding agencies, USDA, NIFA, and CBC grants, and especially one of our master student, Rosalind Battle. Most of the work conducted in her master's thesis, and also Surya Krishna Siktivel, PhD student in our lab, who is going to help us for transformation and also for genome editing. And also Venkat Hemant Limada, he was a graduate student in our lab. Now he is doing his PhD at Purdue University. So with this, I would like to thank Dr. YSR Horticulture University for giving me this opportunity. Also, I would like to take any questions.
Thank you, sir. Thank you very much. Excellent presentation on gray molds in strawberry. And I have highlighted the China is the top most producer of strawberry, followed by USA. and potassium silicate studies controlling the botrytis, that is gray mold, and genetical analysis, that genotypes resistant varieties, and leaf disc method, and genetical genes, what are the genes responsible for the resistance. So excellent presentation, sir. Thank you very much. And now it is open for discussion. Any comments from our university officers, faculty, and students?
Good morning, Dr. Amarnath.
Hi, sir.
Good morning. Good morning. And
Good morning.
very nice presentation. And I have one basic doubt. Do these silicates have any harmful effect on, because we are being used these silicates on this after harvest also on the fruits to reduce the botrytis infestation.
Yes, sir.
Do they have any, harmful effect on the human beings? Because these potassium and other silicates, sodium silicates, they are being used on these grapes and melons also.
Correct.
So,
Yes.
For against these fungal diseases.
Yes, sir. So if you use in the recommended dose, sir, they may not. That is why we are studying different doses. Even if 2 ml treatment is being effective, but if we use higher doses, no use. So that is why we need to educate, farmers and people how much we need to use that. So if people may think, since it is protecting, if you use higher concentration, they may helpful, but if they are not effective actually. In our treatment, if you see, even we sprayed 3 ml or 4 ml treatment, that is not effective. So only 2 ml treatment. So the recommended dose, sir, may not be harmful to the human beings, or that is why we need to test after this again on the humans, like up to what dose of the potassium silicate is not harmful not to the human beings actually. That is, maybe first we do this, then after that the recommended dose will be tested by the food labs. Then only we can recommend to the farmers, sir. That is why we are still not able to we are recommend to the farmers level, but at least in terms of research we are developed this kind of studies. Yes.
But anything alternatively are being, I think you have been studying this gene transfer also for imparting resistance. I think that may add some, value to your research work.
Exactly. Yes, sir. That is why we are yes, sir. It's continuous process we are doing, sir, and simultaneously they are under pipeline. So we wanted to study and find out this whole mechanism, sir, why they are resistant these chemicals, and more than that, we wanted to in the future, we wanted to do multi-omics approach to identify what kind of genes and metabolomes and all kind of things are playing a role for bringing this resistance. And as you mentioned, is there any other off-target effect on the plants also we wanted to study? For example, now we may be thinking that potassium silicate good in terms of protecting from gray mold. Maybe there may be that other cost effective on other traits. So we wanted to study that as well.
Right. Thank you.
Thank you.
Sir, I have one doubt. You studied 150 genotypes evaluated for the resistance. How
Yes,
many found resistant out of 150 in our study?
So out of 150, sir, based on the percentage injury, we narrowed down to I think 40, sir, 40. Then what we did, again, we did validation, sir. Among the 40, then we narrowed down to 10 lines, sir. Those 10 lines are now testing under the field conditions.
Thank you.
Thank you.
Hello.
Yes.
Thanks, sir. It was a good presentation. So it was nice connecting with you, as I'm a PhD student at Clemson University here. So basically doing my PhD in plant pathology. So I was more, curious about plant pathology aspect. So you mentioned that you carried out the greenhouse study for spraying this potassium silicate, right? So, did you check basically botrytis produce sclerotia? It survive in soil as sclerotia, right? So, did
Yeah.
you check if there was any sclerotia in the soil before while you grew the strawberry plants?
So the strawberry, right? What we did so far, we did not spray actually. So on the only up to leaf discuss, like or in the fruits only we did not test on any soil at our whole plant level, not yet.
Oh, so is it like post-harvest spraying of the fruits only?
Fruits only, and also we did on the leaf disc. You saw that leaf disc assay, right?
Yeah, right.
Yeah, we infected only on the discs. So to identify the intracellular level tolerance, so we did on the discs. So now the field evaluations are going on. There also we will not spray any. We will inoculate, but we will check the natural botrytis infection rate on the field. So field, right? We just grow the strawberries. We will not spray any pesticide, and we will like rate them based on that infection rate, botrytis fruit rot infection on the field. So that is why
How,
we will.
how about drenching the soil with potassium silicate? Like if there is any sclerotia and if there is an infection due to botrytis after sclerotia, germinates.
That
So
is
is
also
it?
more effective. Yes, some studies has been done, like they apply to the soil. So
Mhm.
yeah, they have been done, but we haven't not studied.
Okay.
So, but a lot of the people are doing. Yeah, that's also a good approach. Like not only bringing like antifungal activity are on the soil, and also they are helpful for strengthening the plant in terms of root structure, and also silicates play important role in the formation of cutin on the cell layer. So when the cutin of the cell wall, or maybe if it is strong, the pathogen entry also will be low. That is what some studies shows, that silicates are very important in terms of protecting both environmental stress. If cutin layer is there, even you can evapotranspiration transpiration of like water evapotranspiration also, like can be reduced.
So how much percentage reduction was there in the disease after you sprayed the fruits or you conducted the leaf disc assays?
So percentage, right? Percentage reduction.
Yeah,
Yes,
like percentage
if you see in
reduction
that
in.
graph, I think more than 50 to 70%, right? So in the 2 ml treatment, not with the all other treatments. Yeah.
Okay, thank you. It was
You're
nice
welcome.
connecting with you. Same picture.
Thank
Thank
you.
you. Thank you, Priyanka Gupta, that you're the PhD student and a graduate research assistant at Clemson University, South Carolina.
Yes.
Thank you for connecting Global Connect series of Dr. YSR Horticulture University.
Thank you, sir. It's a good initiative that you people are carrying out. So it's very helpful for us even. So thank you so much.
Thank you. Thank you. Anybody from our university officers? Yes,
Sir, am I audible, sir?
yes, sir. Yes. Proceed.
What exactly is the mode of action of this potassium silicate? Is it preventing the entry of the fungus because the silicate is forming an external layer and it is preventing the entry of the fungus, or is it any other mode of action?
So that is where, sir, we are still like not confirming that disease resistant how it is bringing. That's why we are doing more studies. Some studies shows that because that role is there in terms of strengthening the plant structure, and also when we studied the in vitro study, you can see that even that antifungal activity also is there. So that is why more studies are needed to confirm the function of that potassium silicate effect on the plants, actually how it is bringing in terms of so that. But even not only in our study, some studies also shows that it has some kind of antifungal activity.
Even in crops like rice, the varieties which are having high silica content are more resistant to diseases, sir. So they look
Yes,
for
sir.
more high silica content. Is the same mechanism working here?
Like that only, sir. That is what we were like now curious to know. This is the first reports are even in the strawberry, right? Even though they reported it will give resistant like that, but in companies, but nobody studied uh, like comprehensively, like what is the mechanism. That's why at least we started with some genes now. at least because of the potassium silicate transporters. Because of, sir, once maybe that's also a good point. Maybe it's preventing the entry one way, and both functions may be there. That is why it is becoming more entry and also antifungal activity. So that is why it is more effective compared to others. Like that is what we are like assuming. But to say that, we need like more molecular studies, sir. So that's why we are like now doing that part also. But so far, this is our results. But we are underground. We are still digging to understand that mechanism actually.
Even the results are very surprising with just at 2 ml, it is working very highly, like highly antifungal. With just 1 ml extra concentration, the results are counterproductive.
Yes, sir. Yes, sir. That's why we are first we up to like we ended our study with only like up to proving that strengthening parts. But later, since this 2 ml treatment showing good results, we continued like we wanted to dig like identify why it is like will this effect will be there on the fruits also, like that we studied, sir. Like actually the initial that field studies was done in 2021 and 2022. Then this fruit assess we did it in 2020 last year, actually 24, because of this curiosity only. But luckily it worked. So that is why before sharing this or publishing, we are confirming within ourselves, like, so that's why we now at least we are in a stage to publish, sir, this article. Yeah.
Did you record any other disease incidents or severity like this, any scale?
Actually, yes, sir. We are also that in the field study, they are doing even also anthracnose.
Okay. Thank you, sir.
Thank you.
Thank you, Kiran. Next.
Maybe any questions from students, sir?
Any student, any teacher?
Another thing, you use a lot of molecules, but surprisingly, like, are they not effective, or like the pathogen is developing resistance? I could see many advanced molecule like strobilurin based and other molecules. Are they, how far they are effective, or like over a period of time they are losing their resistance?
Which
The-
one, sir? You mean in the terms?
You said like, you tried many chemicals, right? That includes many advanced strobilurin based fungicides and other molecules.
Yes, yes, sir. Yeah.
So are they completely lost their efficacy, or like they are only partially effective? Did you?
They lost, sir, actually the dose, recommended dose. For example, now before they were spraying 1x dose, now they are resistant up to 2x, 3x. So that is why they are like spraying more concentration. That is becoming harmful to the humans and also environment. So that is why now people are recommending, instead of spraying one pesticide, spray in the combinations or go for the alternatives, or grow like organically. So that is what like now recommend. As last lot of people losing interest on consuming because of the pesticide. If if you take the data in US, even though they consume a lot of vegetables, fruits, but inflammation is becoming very major issue in terms of developing cancers and also other obesity and all those things. So one reason they are saying that high usage of pesticides in the fruits, like. So yeah, that is why like recommendations are going in different way. Let's see, sir, how it works. Yeah.
క్రాంతి కుమార్, మొన్న ఇది తీసుకున్నారా? ఇది స్టెబిలైజర్ బిల్ తీసుకున్నారా?
Anybody? If anybody wants to ask questions, you can pass.
So also I would like to share with students one more thing, sir, if they are okay.
Yes, sir. What is the scope and the job opportunities in our USA for horticulture students? Just another my query. And our Honorable Vice Chancellor is very much interested to sign MOU with Delaware State University. And so kindly respond, sir, in that. And job opportunities for the MS and PhD students, and the faculty exchange and the faculty.
Yes, sir.
Kindly
Sure. Sir. Yeah, sure. So yes, sir, when it first come to student thing, so I would recommend people to follow this website, American Society for Horticultural Sciences. So if you go here, the career resources, so there you will see a lot of advertisement for graduate students. If any lab, any like people are have any vacancy for a graduate school, so they will be posting here. So you can go through this website actually, especially for horticultural sciences. If you are looking for PhD or postdoc positions or any other fellowships, or any conferences, information will be there. Also, you will have society journals, 3 journals in this. So this is like really good place to check, and you can join as a member, or like sometimes they will give student for the students they will give travel grants, so they can apply for those. So you will be like getting all the information from here. And similarly, we have all Crop Science Society of America, American Society for Plant Biologists, ASPB. So similarly, like all societies, it's really good for they will advertise all kind of information in terms of post and also schools. Maybe in the couple of like sir, as I mentioned to you in upcoming meetings, maybe I can organize 2 to 3 students to share how to prepare or how to apply for universities in the United States, and how to write cover letter and how to approach professors on those informations. As I mentioned, maybe that will be a good source for all the students to learn. Maybe that will be more focused toward the students. And sir, coming to the memorandum, sir, I discussed with our professor and also head of the department. So currently now, due to this election and recently this new government and also, they asked me to wait for a couple of more days. If anything like any news, I will get back to you, sir, on those. So yes, the, coming to the opportunities, sir, US is the like land of opportunities they call. Yes, we have a lot of opportunities. So they also look for students, like especially this horticulture and agriculture universities are in India. They are based on US syllabus. So they highly prefer, sir, your Indian students if they have like good in terms of uh, percentage, and also if they can do able to do good research, and if they published any papers they consider. And also these days they are looking for so actually scores are actually either TOEFL and GRE. So score. So those two are very important in terms of to students to get graduate research assistantship. So maybe now that one student from Clemson University joined, and 2 more students from our university, and also one top like most university like Purdue and Cornell. I can ask some students to give guidance on for the students how to apply and how to get into the schools and jobs, sir. Maybe that will be useful actually.
Thank you, sir. Definitely, we will get feedback from the students and let them share their experiences going to America from India. Definitely, we will arrange one Zoom meeting on that. Thank you, sir. And if nobody is there, we'll go for a vote of thanks by Registrar Dr. V. Srinivas garu, Dr. YSR Horticulture Minister.
Thank you, Dr. Dhananjay Rao garu. I immensely thank Dr. Amarnath garu for your excellent presentation on the topic today, enhancing strawberry resistance to gray mold through fungicide alternative studies, advanced phenotyping and genetic research. On behalf of our Honorable Vice Chancellor, Dr. K. Gopal garu, and the program coordinator, our Director, Industrial and International Program, Dr. Dhananjay Rao garu, from all our university officers and faculty, associate deans who have participated in this program. On all their behalf, I extend once again sincere thanks to you for your very wonderful presentation on this topic with really a quite, colorful presentation of your slides. As beautiful as strawberries.
Thank you.
Yes, very, very beautiful presentation with a lot of technical information. I think it is very useful to our staff also, because strawberry is not being extensively grown in India. But the technology what you have developed there will be mostly useful for the other crops also.
Correct.
Like
Yes.
melons, goats, where this botrytis problem is high,
Common,
this
yes.
fungal problem at both pre-harvest and post-harvest stages. I think whatever the work you have done definitely will have a great impact, I think, to develop some good mechanism for developing some varieties for resistance towards this one of the major fungal disease that is causing havoc in American country.
Yes.
What the technology you have developed definitely will be fruitful in coming days. I think you will come up with some good recommendation. Thank you very much for your very nice presentation and sparing your time for being with us for almost 1.5 hours.
Oh, thank you, sir. It's my great pleasure, and I'll thank Dhananjay Garu and also VC and sir, you register, sir, for this opportunity.
Yes,
And
yes,
thank you. And also Padma madam mentioned actually
yes,
about you, Padma.
yes, yes.
Yes.
And also extending our thanks to Priyanka Gupta, student. So she has also joined this program. And also you have given some, I think, some good scope, I think, for us to have some MOU with your esteemed organization.
Yes.
And there is also a good scope for these fellowships and research work. We have combined, I think, the program for this American Society for Horticulture Sciences,
Yes.
and also for Society for Plant Biology. I think you
Yes.
have given.
Yes, sir. Even any students, sir, if they wanted more information, they can reach out
Yes,
to me.
they can see this website.
Yes.
I think a lot of information will be available, which will be useful for them also, for the students who have, opting for higher studies,
Correct, sir.
for any research grants and all. Thank you for providing very good and useful information. Right. Thank you. All the best.
Thank you, sir.
Thank you, sir. Thank you, Registrar Sir, Dr. V. Srinivas Garu. And actually, our Honorable Vice Chancellor, I want to have MOUs with international reputed esteemed institutes, and we are planning to have MOUs. And sir actually wanted to join, but due to some emergency work, other work, he could not join. Thank you, sir. Thank you, Dr. Amarnath Reddy Garu, for accepting our invitation and nice presentation, excellent presentation. So thank you one and all. So
Thank you,
now
sir.
now
I
the
will be in
program
touch.
ended. Definitely will in touch, sir. So any student, if anybody will ask, definitely will in touch with you, and I will forward. Thank you once again.
Thank you, sir. Can I leave the meeting, sir?